This next Zoom section may not be as engaging for a viewer as it has been for me, because since 1985, since I got involved in this, what turned into nutritional research that started out to be a middle-of-the-road scientific effort to register a new drug with the Food and Drug Administration that you know as the FDA, to have a major publication sent to you that supports everything that you have observed. And there have been multiple anecdotal examples and pilot publications that are cursory but not formal, that to have something published in China, paid for by the government of China at one of their institutions, that is a 43-page document, as I hold up in my hand, that's entitled "A New Biomaterial Derived from Aloe Vera dash Acemannan from Basic Studies to Clinical Application." This 43-page document is virtually, I would say, a Bible on the active ingredient from aloe, that is independent validation of things that I never had the research funds to prove properly in the most formal manner are present. In addition, the bibliography is awesome, that it's over 100 papers from multiple sites of multiple investigators that have published papers that they give a citation in addition to the work they did in China. In addition, I hold in my hand another document that is from organizations in United Kingdom, the British Society for Allergy and Environmental Medicine and the British Society for Nutritional Medicine, which is a position paper sent to the National Health Service that is totally modern medicine, conventional medicine oriented for the population of the United Kingdom. And they're urging them to this group to look into nutrition for its benefits for the population, for being safe, effective, and economical compared to drugs. So these two papers are vitally important in terms of supporting information that literally I've had qualified people basically saying I'm nuts in terms of what we've been able to see and prove with money that Carrington Laboratories paid for research to be done in first 10 and then 12 academic institutions that were generating basic science and clinical studies for the value of the active ingredient from aloe vera. So I'm going, in this lecture, I'm just going to go through this 43-page document from China, paid for the Chinese government, that validates and supports activity that I've been involved in now since 1985, or 39 years ago. Now we'll go to the first slide. Now, this paper that we will superficially review was paid for by the government of China and one of their institutions. And we'll go to the title of this paper, A New Biomaterial Derived from Aloe Vera, Acemannan with Basic Studies to Clinical Applications. And what's in this paper is so incredible from these 4 Chinese investigators, which they're at the School of Somatology at the Medical University. But as you look into the details of the research, it sounds like it's more of a dental school, but in some of the foreign countries, the doctors and the dentists and others, first 2 years of academic training are pretty much together before they go into their clinical junior and senior year. So I don't know whether that's what this is, but you can see the 4 Chinese investigators that worked on this paper. And it's just amazing to me to see this validation, which I feel like that our population needs to understand how important the nutrient molecules in the natural plant kingdom support the engineering and design of life that is in DNA in our chromosomes. And part of the really amazing thing about this program in DNA is it controls cellular synthesis on a little assembly line in every cell in the body, where nutrient molecules, including elements and vitamins, together are hooked together to make bioactive compounds that carry out the biochemistry and physiology of life. And this paper from China is amazing. Now, this is the abstract of the paper, and I've not too good at making lines, but I wanted to point out that they say it's a novel biochemical material in tissue regenerative medicine, is that first green line. And then it goes on to say that regeneration engineering and soft tissue repair, among other operations, are present when you add aloe acemannan to tissue cultures and to the diet. And very important in this is something we saw a indicator of it back in the 1980s at Carrington Laboratories that was paying for research to be done to try to get through the FDA approval process for a new drug, is that the active sugar in aloe leaf gel is mannose, but it has no activity without the acetyl group. And in fact, they do a series of experiments in this paper showing that the activity and the functions of the mannose varies with the degree or percentage of acetylation that gives it its bioactivity and functional properties. But then it is the key words I think are significant. As it says that there's tissue regeneration, dental regeneration, bone regeneration, wound healing, immune regulation, and anti-neoplastic, and that means anti-malignant cell activity and antiviral activity. And I might add to that antiviral activity, it's a different type of approach. The acetylated mannose does not attack the biochemistry of the virus or of a bacteria or fungus or tuberculosis organism or even blood parasites like malaria. All what it does is boosts the body's host defenses, both cellular and humoral, against any infection. So these are key words that are in this paper with its thing. Now, the paper goes into, as I said, the degree of acetylation of this mannose that is made in the leaf of the aloe leaf gel. And we've got on the left-hand side of this slide, different molecular forms of the degree of acetylation. And it points out that there's different chemical environments that the amount of acetylation influences its bioactivity. And in this paper, they give you photographs of the different ways that you process aloe acemannan for different applications, which are very broad, and that depending on whether it's a powder or a gel or a film, which can be done in the processing, they go through all these permutations of processing. And what you end up with as the acetylated mannose for the this is the reason I say this is almost, I'd say, a Bible for the properties of aloe acemannan. And in the basic science publication, they go through the different processes that it can be either a spray, which I'll add to the films or the spongy particles that adhere to mucosal surfaces if they're moist, and that they point out that the degree of acetylation determines how effective the aloe acemannan is in different clinical applications. Now, one of the things that this paper points out that the biological function is it's an immune modulator. And in the early stages of Carrington research in the 1980s, we thought we had the most incredible immune boosting molecule from the aloe plant. And then we heard from people that had overactive immune systems, like in asthma and some of your autoimmune diseases. And they were getting better too. So here we had overactive immune system and then, shall we say, lazy immune systems. And I actually went to the trouble to go to a conference in Europe for a week where they just studied and spent a week on the immune regulation. And we realized we didn't have a stimulant or a suppressor, but a modulator, that if the immune system was overactive, it would bring it down to, shall we say, rest or the normal zone. But if it needed to be activated against an infection, it would increase the activity of the immune system. And of course, at Texas A&M particularly, but other institutions too, every virus that we could find that is available for animals as well as humans, that there was apparent antiviral activity, but it really was boosting the body's host defenses against any type of infection. And then studies were done at Texas A&M that they had a survey model from the National Institutes of Health called a mouse tumor that was notorious, that no activity whatsoever had been done, that it was 100% death rate. And by providing the active ingredient from the aloe leaf gel, 41% of the animals destroyed all evidence of this survey tumor, to the amazement of the investigator. And they continued to do research on this. That was with one injection that IP, that they got 41% destruction of the tumors. But with twice a week injections, they got up to about 67%, and combined with removing the tumor surgically like you generally do in humans actively with a neoplastic or cancer, that the combination of surgery and the active ingredient of the aloe plant got up to about 90% survival of the animals in this survey model. And this was so dramatic that the paper submitted to be published was rejected by the individual, saying it just couldn't happen for this model that was 100% fatal to get this kind of a benefit. In this paper, they have multiple dental applications because there are different kinds of bones in the skull and in the jaw and in the where the teeth are in the malar ridges. The bones have different properties, and this application of the acetylated mannose regenerated bone of all types. And also teeth have different layers of enamel and cementum and other materials to develop this hard tooth. And the acetylated mannose increase the formation of these different layers of the tooth. And then there was a paper that oftentimes there's an abscess of a tooth and up into the bone. And they pointed out, it seemed like when I read the article, that rather than doing a metal post that's trying to sterilize the tooth and do a root canal job, that this material regenerated the pulp inside the tooth, and the research that could be done for vibration, cold, and hot and all, that the tooth was viable again. And I sent this to a dentist to see if he came to the same conclusion that I did. And while waiting for a response, I got a telephone call from an individual in Houston, Texas, that he had gone through this process, and just to reduce the pain from this abscess in the tooth and in the bone, he had put directly the aloe Acemannan on the gum several times a day and at night, and it reduced the pain. But they scheduled in the future when they would come in and do the root canal, and by the time they got it, he had no symptoms, and they re-x-rayed and all, and that the abscess in the bone and at the regeneration of the pulp, that the tooth was like a normal tooth again. So this confirmed the research activity of this paper. And then they point out that soft tissue of the gums regenerate. And I had stated before, in that bone, they have several experiments in this paper from China about the different types of regeneration of bone if you provide acetylated mannose in the environment of the bone. And that this soft tissue healing is one of the oldest things in history. Fact is, it's stated that Alexander the Great, in his war train, had large containers with aloe leaf growing, and that they used the gel from the leaves for sword and knife and spear wounds, and fostered healing with less scarring and reduce infections in this primitive warfare. And this was just confirmed by this Chinese investigation. Now, this to the right is different examples that they had in their paper of actual experiments they did and the values they got. And that if you read from left to right and down, that the aloe Acemannan modulates macrophage activity, a big white cell, and it enhances cytokine production. Now, cytokines are molecules made by the macrophages that act between cells, much like sending a message that used to be like cards in computers before we had typing keys. And these molecules carry information, which are cell activators. And these macrophages, with addition of the aloe, increases the amount of these molecules that are sent, not only from different cells like fibroblasts and mucosal cells, but dendritic cells in the nervous system. And where there's needed, there's suppresses macrophage activity, and it suppresses the activity of T lymphocytes, and it increases the macrophages producing nitrous oxide, which is used to dilate capillaries to increase the blood supply to the cells. And that interferon plus the Acemannan increases the nitrous oxide production. So what they also showed in this experiments to the right, that you could take mice and irradiate them till you destroyed their bone marrow. But if you gave them the acetylated mannose, they would repopulate their bone marrow and recover from what would be lethal radiation. And of course, this has a great potential in human radiation for malignant cells that is done. Now, in addition, again, these experiments that they actually show you their results on different pages of this article, that there's enhanced host response to any virus infection, that this acetylated mannose causes cancer cells of the GI tract or the colon to die. That's what apoptosis is, the dying of malignant cells. That they have the experiments that the pulp of the tooth is regenerated, as I mentioned before, in these teeth that they usually put a sterilized pin in to try to keep them in the mouth. And lo and behold, the acetylated mannose regenerates the pulp. And the infections of the jaw and the teeth are cleared. And they show experiments that the different layers of the tooth are enhanced to reform and make stronger teeth. The experiments at the right show different types of bone formation models that they had, that the bone, depending on different types of bone, work better. That where there's tooth extraction, that the bone defect where you pull the tooth out is regenerated, and that clavarious bone is growth is increased, and alveolar bone and periodontal soft tissue growth around the bone is increased, and cementum for the structure of the tooth is increased proportional to the concentration of the acemannan that is provided in these experiments shown at the right. In this 43-page publication from China, that they show that the acemannan increases collagen production and fibroblast activity for healing of wounds and regeneration of soft tissue, particularly around the teeth and gums. Again, we talked about cytokines being like IBM cards, molecules to carry information. Well, the interleukins are another type of cellular communication molecule. The interleukin 6 and interleukin 8 are increased, and that there's increased antibodies that can be detected in the tissues. And as we have said before, that oral wound healing is accelerated, and very important, it accelerated the healing in diabetics, which is a great problem to diabetics, that the high blood glucose interferes with the metabolism of the fibroblasts and white cells, and being able to handle infections, and that non-healing ulcers of the legs and the back or bed sores that happens in the diabetics, that there's accelerated healing that you can't get with just a good diet alone, that this topical acemannan increases the healing rate. More dental experiments were done, and we have mentioned the dentine pulp regeneration, and they point out that children's deciduous teeth are formed more rapidly and stronger when they finally come in, and that the layer of enamel that protects permanent teeth is increased in the amount and the thickness by providing acetylated mannose, and that the root development of the permanent teeth is enhanced with in the children that they include Acemannan. Now, the general mechanism is that the cells send molecular signals from, say, macrophages to effector cells like fibroblasts that respond to restoration of deviations from normal tissue environments. In other words, the genes are activated to restore normal structure and function within the soft tissues and bone and the teeth. And again, this emphasizes the importance of nutritions to support the code of life that is in DNA or our genes to make bioactive compounds structure and functionally, that this information is in the 46 chromosomes in every cell, and that the programs in these genes or in DNA are for normal structure and function, or balance, which is an old subject that Hans Selye, the father of physiology, called homeostasis, or balance of the metabolism in the cells of all organ tissues. And so aloe acemannan and other phytonutrients that we have added that are low or missing, or by choice that people in the United States tend to not include in their diet, are put together in what I have developed over the years, what we call New Eden, which is built around this acetylated mannose to foster all these benefits that this independent validation from this research in China supports. But more importantly, I think, is that the bibliography has over 100 papers from different sites and different investigators in different countries that have done different experiments that validate the anecdotal observations that were reported to me over the years. Now, in this research from China, this is just a categorization of these different pages of the published papers. And though you can't read them with this light printing, these papers are divided into different categories of benefits that validate anecdotal observations that I never had the funds, because the goal of Carrington Laboratories was to develop a new drug against the AIDS virus, and that's what they wanted to spend their money on, and they didn't want to chase all these different benefits that these investigators have found that the aloe acemannan supports. And these topics that they are divided into are antiviral, anti-neoplasia, or the neoplasia is new cell growth that gets into the malignancy area, multiple dental problems, tissue and bone regeneration, different types of tissue and bone, and wound healing is accelerated, and even the balance of the metabolism for handling blood sugars in diabetics, and the lipids that lead to atherosclerosis, even they were changed to be more normal and to reduce the blockage in the arteries of these different papers that have been published in these different countries. So the summarizing all these papers, I adds that 90% of these papers have been published since the year 2000, And on the second page of this article, they cite the key papers that our research group developed at Carrington Laboratories financing in the 80s and 90s. And that was very gratifying that they acknowledge this work before these papers are listed in the bibliography. But there are some points that they were not aware of, apparently. Number one was that the new drug application by Carrington Laboratories was rejected by the FDA, whereas in their paper they say there should be activity to make sure that this substance from the aloe can be entered into a new drug process to be part of standard medicine. Well, the FDA rejected because no toxicity whatsoever could be found, and you couldn't give them a lethal dose that kills 50% of the animal or the lethal dose that kills 100% of them. There must be a wide window between 50 and 100%, or they won't let you proceed. Well, of course, with the FDA, the biggest problem was we couldn't give them LD50 or LD100 because couldn't kill anything, no animals, and even 3,000 milligrams a day taken by undergraduate students at the University of Texas in Austin, and even in 50 medical students that had sterile Acemannan injected into their vessels, didn't kill a medical student. And all they did was boost their body's defenses against any type of deviation from normal. And the second big problem with the FDA was we got reports of benefits, whether the deviation from normal is physical, mental, or emotional. And I had not really realized before this that medicine has a mantra that there are no panaceas. And this activity of the aloe acemannan exceeds any type of panacea or something good for everything that repeatedly was anecdotally observed as it was being utilized by large numbers of individuals. And the next thing that I wanted to point out is that a dietary supplement company that I was medical director for a decade sold over $5 million of Acemannan as a key ingredient in dietary supplements because of all the benefits. And the biggest thing was that you should make no claims, anybody that was distributing this, to treat or cure a disease or abnormal condition, but the individuals violated this. And so the company ended up paying a $10 million fine to the FDA because of what the distributors did, which was even worse. They claimed that I was told, for example, they would say I was told I was going to die from this condition. And I took this dietary supplementation, and the medicines had done no good, but now I'm normal and they can't find anything wrong. And they told them, if you do what your doctor tells you to do, you'll probably die. Well, that violated not only the federal restrictions against claiming natural products or nutrition could support normalization of deviations from normal health. It violated state professional laws. And the very important thing that was found out is we have the research paper that 9 molecules of this active ingredient from the aloe plant, acetylate mannose, in the endoplasmic reticulum, which is the second major step in cellular synthesis, these 9 molecules and 3 chains make a domain on the membrane of all cells of the body, which is a domain for balance and normal activity, which has been identified as homeostasis. And in homeostasis, when everything inside the cell is moved toward the normal zone, which is the program in the DNAs, you don't have deviations from normal, which for communication purposes in medicine are called diseases and syndromes. And this is where the big conflict develops between natural nutrition and the drugs. Because drugs interfere with the engineering and design of life to suppress symptoms. Drugs do not address the cause for the symptoms, whereas the natural nutrients support the engineering and design of life as is coded in DNA to correct the cause for the deviations from normal and eliminate the cause for the symptoms. So that's the power of these natural molecules found in the plant kingdom. One of the things we found out is that the acetylated mannose, when taken, generates millions of pluripotential adult stem cells, which on flow cytometry are CD, stands for cell domain 14 monocytes. That's from the bone marrow, your own cells, and you have no problems of rejection like you have by donation of stem cells from any source. That the regenerative and support for a normal physiology is incredible by these stem cells that migrate into tissues. And this is why we have had reports of individuals that were on organ transplant lists for replacement of their hearts, their liver, their kidneys, their lungs, that by taking the oral Acemannan, that these stem cells migrated into the tissues and restored function to the point they were no longer qualified to have one of these rare organ transplants where it's an organ from someone else and the immune signature on the cells is foreign and you have to take immune suppressant drugs. Well, when your own stem cells come from your bone marrow, you don't have to take suppressant drugs, and they have incredible power to regenerate function structurally and functionally. Also, that the acetylated mannose was given to Alzheimer's patients at the Miller School of Medicine, and memory and cognition in these patients was increased. And in fact, the best responses were in some of the worst dementia patients. This has been published and is in the literature. And it should be known that this voice that you hear was invited to come to Canada because they were having a big conference in fetal alcohol syndrome. And they wanted me to come and explain the activity of acetylated mannose. And I was, quite frankly, overpowered with finally some experts, you might say, wanted to hear what I had to say about the ability of acetylated mannose in biological activity. And I found out the reason they invited me is that they had fetal alcohol syndrome damaged children taken away from their alcoholic mothers and placed in foster homes. And they had 7 of them that their average IQ was less than 50. They were not making any progress in special education, and they were behavior problems in school as well as in the foster homes. And all of a sudden, they began to improve on their activity. They were behaved better, and they began to improve their academic performance in their special education. So they repeated their IQs, and they found out they ranged from 80 to over a 100. This is a range from an average to a individual that probably could do well in college, that before with the IQ less than 50, they were destined to be wards of the state for life and never live independently. And they wanted me to explain this. Well, I have written a fascicle about how the process we went through to discover and find and prove that the acetylated mannose generates millions of adult stem cells, and it's primarily done with flow cytometry that follows these stem cells. In addition, I might add that a paper was published in the year 2000 with a researcher that objective biomarkers associated with the disabilities of aging, which was developed at Tufts University and Johns Hopkins at that's Boston and Maryland. The reason this was done is after World War II, the federal government found out that people were living longer and continuing to get assistance from the Social Security Administration. People just weren't dying and continuing to get benefits. And so their study of these two universities was, what do we mean by somebody is getting feeble and old? And they developed objective biomarkers that could be measured and provided them. And that's what we used in this study of 600, over 260 individuals that were over 50 and had given up various mental and physical activities because they were getting older. Well, by adding the acemannan to their diet, apparently these stem cells migrated into their tissues and regenerated muscular activity and also brain activity. And they returned to physical and mental activities they had given up 5, 10, 15 years before. And now this, we have one of those individuals was a dentist from Wichita, Kansas, that said, "This is incredible. I'm 72 years old and I'm doing so well." He had his telomeres entered, assayed, and telomeres are caps on the ends of chromosomes, which protect the chromosomes from damage. And actually, it's known that telomeres are longer in people that live to be 90 to 100 or older, and they're short in families that somebody dies usually between 55 and 70 from some chronic disease. And also, their telomeres are short in animals with short lifetimes and long in animals with long lifetimes. Well, the aloe acemannan, which they didn't mention in this paper, elongates telomeres. And to add routine exercise to having this in your diet is synergistic. And though this study is ongoing, about halfway done, this is incredible benefits that tell why those individuals that increase their biomarkers associated with the disabilities of aging, it was because their telomeres are lengthened by the effect of acetylated mannose. So the ability and this independent validation, not only by research conducted in China by their government, the papers that are in the bibliography from multiple sites and individuals proving the benefits clinically, and basic science studies too, that acemannan has incredible regenerative and restorative effects on the human metabolism is absolutely incredible. So that's the last of that paper that I want to give you from China. And I've since found out that in Iran, that they have done a number of papers, and it seems like these two countries that are in competition or antagonistic toward the United States are following technical developments that gets published in our journals better than our own country. And one of the reasons is that more comprehensive nutrition is It's destructive technology. It threatens the drug paradigm that human beings make chemical molecules that are poisonous to the engineering and design of life to hopefully reduce symptoms. And so nutrition is utilized by the genes in cellular synthesis to move the body's metabolism, structure, and functionally to the normal zone, and this independent validation from China supports this.